Hello friends, welcome to my educational YouTube channel Quest for Knowledge. Today we are going to discuss about autotrophic nutrition. In this type of nutrition, the organism is able to perform the process of photosynthesis. Autotrophs are able to utilize the materials from the environment and convert them into energy. So let us discuss the process of photosynthesis. In this process, green plants take in substances from the environment and convert them into energy, which is stored in the form of food. The materials taken from outside are carbon dioxide from air and water and minerals from the soil. In presence of sunlight and chlorophyll present in the leaves, they convert the raw materials to carbohydrates, which are used for providing energy to the plants. Carbohydrates, which are not used immediately for release of energy, are stored in the form of starch, which serves as the energy reserve to be used as and when required by the plants. During photosynthesis, oxygen is given out as the byproduct. Let us look at the states of photosynthesis. Plant cells present in the leaves contain chloroplasts, in which chlorophyll is present. Chlorophyll absorbs sunlight in the form of light energy. Stomata is present on the surface of the leaves. Carbon dioxide enters through the stomatal pores when they are open. Water and minerals are absorbed by the roots, which reaches the leaves. During photosynthesis, light energy is converted to chemical energy. Water molecules split into oxygen and hydrogen. Carbon dioxide is reduced to carbohydrates. Overall chemical reaction of photosynthesis can be written as 6CO2 plus 12H2O gives C6H12O6 plus 6O2 plus 6H2O. We discussed that green plants, they use raw materials from the environment like carbon dioxide and water. We also discussed that sunlight and chlorophyll is necessary for photosynthesis. Now let us perform an activity to see that chlorophyll is necessary for photosynthesis. Aim: to show chlorophyll is necessary for photosynthesis. Materials required: potted plant with variegated leaves, white sheet of paper, pencil, beaker, test tube, forceps, alcohol, iodine and water. Procedure: take a plant with variegated leaves. For the activity, I have selected a potted plant with variegated leaves. Keep it in dark room for 3 days. I have kept this plant in a dark room for 3 days. Place the plant in sunlight for about 6 to 7 hours. I have kept the plant in sunlight for 6 to 7 hours. Pluck a leaf from the plant. I have plucked a leaf from the plant. Trace the outline of the leaf on a white sheet of paper. Mark the green areas containing chlorophyll. I have drawn the leaf on a sheet of paper, marking the green areas present on it. Dip the leaf in boiling water for a few minutes. I am taking the leaf and boiling it for few minutes. Immerse it in a beaker containing alcohol. I have taken alcohol in this test tube and now I am going to put the leaf in this test tube. Boil it in water bath till the leaf decolorizes. Now I am going to take this test tube for water bath. Here you can see that our alcohol has started to boil. Dip the leaf in iodine solution for few minutes. Now after the water bath, I am going to take out the leaf from the test tube. So here you can see that the chlorophyll present on the leaf has dissolved in the alcohol. You can see here that this leaf has become colorless. I am taking iodine in this beaker. So I have taken iodine solution in this beaker and now I am going to immerse it in iodine. Take out the leaf and rinse off the iodine solution. Now I will rinse the leaf in water. So I am rinsing the leaf in water to remove excess iodine. Observe the change in the color of the leaf. Observation. The leaf has two types of patches, bluish black and yellow. The bluish black areas contain starch and the yellow areas are without starch. The bluish black areas are the ones which were green previously. Non-green areas of the leaf remain pale in color. Conclusion. Chlorophyll containing areas which are green in color prepare starch, which is the product of photosynthesis. Therefore, chlorophyll is necessary for photosynthesis. So we can conclude that chlorophyll is necessary for photosynthesis. Let us perform one more activity to show carbon dioxide is necessary for photosynthesis. Aim. To show carbon dioxide is necessary for photosynthesis. Materials required: potted plant, potassium hydroxide, Vaseline, plastic bottle with cork, beaker, test tube, cutter, forceps, alcohol, iodine and water. Procedure. Take a plant and discharge it by keeping it in dark room for 3 days. For the activity, I have taken a potted plant which I have discharged by keeping it in dark room for 3 days. Take potassium hydroxide in a bottle with a cork. make a slit on the cork. I have taken potassium hydroxide solution in this bottle and I have made a slit on the cork of the bottle. Gently insert one of the leaves of the plant inside the bottle through the slit without breaking it. Seal it with the help of Vaseline. So now I am going to insert one of the leaves inside the bottle through this slit. So as you can see that I have inserted this leaf through the slit. And now I am going to close it. So with the help of this Vaseline, now I am going to seal the slit. Place the plant in sunlight for about 6 to 7 hours. I am going to keep this plant in sunlight for 6 to 7 hours. Pluck the leaf from the plant which is inside the bottle and label it as A. Pluck one of the leaves of the plant outside the bottle and label it as B. 7 hours has passed since I have kept this plant in the sunlight. Now I am going to pluck the leaf which is inside this bottle. Now I will label this leaf as A. I am going to pluck another leaf of the plant and label it as B. Dip the leaves in boiling water for a few minutes. Now I am going to boil these leaves for few minutes. Immerse the leaves in separate test tubes containing alcohol. So I am going to take the first leaf and immerse it in alcohol. Similarly, I am going to immerse leaf B in another test tube containing alcohol. Boil the leaves in water bath till the leaves decolorize. I am going to take these two test tubes for water bath. So first I am taking leaf A. Here you can see that our alcohol has started to boil. Now I am taking leaf B for water bath. So the alcohol has started to boil. Dip the leaves in iodine solution for few minutes. Take out the leaves and rinse off the iodine solution. So first I am going to take out leaf A. Here you see that our leaf has become colorless and the chlorophyll which is present on the leaf has dissolved in alcohol. Now I am going to dip this leaf in iodine solution. And now I am going to rinse off the excess iodine present on the leaf with water. So I am rinsing off the excess iodine in water. Here you see that there is no change in the color of the leaf. Now I am going to take out leaf B. In this case also, our leaf has become colorless and the chlorophyll present on it has dissolved in alcohol. Now I am going to dip this leaf in iodine. Now I am going to rinse off the excess iodine present on the leaf. Observe and compare the change in the color of both the leaves. Here we see that leaf A has no change in color, whereas our leaf B has turned bluish black. Observation: the leaf outside the bottle turned blue-black in color. The leaf inside the bottle containing potassium hydroxide solution remained pale in color. Conclusion: the leaf outside the bottle prepared starch by the process of photosynthesis because it got carbon dioxide from the air. The leaf inside the bottle could not prepare starch by photosynthesis because carbon dioxide was absorbed by potassium hydroxide kept inside the bottle. Therefore, carbon dioxide is necessary for photosynthesis. We discussed about autotrophic nutrition and photosynthesis. We also discussed about the steps of photosynthesis and overall chemical reaction of photosynthesis. We also saw that chlorophyll and carbon dioxide is necessary for photosynthesis. I hope you enjoyed today's video. Don't forget to hit the like button and subscribe my channel.